All right, hello everyone. Welcome back to our next lecture video. This lecture is going to be over the renal system / urinary system. So we're going to see how we create urine in the body. So our urinary system is going to be mainly from the kidneys down. The main structures of the renal and urinary system are going to be the kidneys. We have two, right and left. The ureters, which are going to extend from the kidney down to the bladder. Then we have the urinary bladder, which is going to store the urine until it is full and it is released through the urethra, the tube that will exit from the body. Blood supply of the kidneys comes from the renal arteries and renal veins. We have two renal arteries coming off of the aorta. We have two renal veins which come off of the inferior vena cava. On top of the kidneys, we have the adrenal glands. These are going to be pretty negligible in urine formation. However, they do dictate whether we have reabsorption of things like electrolytes in the form of secreting hormones. All right. We have hormones such as aldosterone, renin, angiotensin system that's going to allow for reabsorption of electrolytes. And that secretion comes from the adrenal gland. So our structures, we have our renal arteries, two renal veins, kidneys, ureters. Two ureters empty both into the urinary bladder, and then there's one urethra which moves the urine out of the body. When we have elimination of that stored urine, so the urine that is in the bladder when it is released, that is known as the micturition reflex. Okay, so urination is actually the process of creating urine in the kidney, whereas micturition is the process of releasing urine out of the bladder. The two kidneys location are going to be in the retroperitoneal cavity, meaning behind the peritoneal cavity. The peritoneum is the abdominal cavity where we have all of the organs stored there, like the intestines and the liver, the pancreas and whatnot. Two kidneys are actually going to be retroperitoneal to that, outside of the peritoneal cavity, meaning that they are not inside of the peritoneal serous membrane. So they are actually outside of the serous membrane, and they are covered by retroperitoneal fat. The ureters are going to be pretty decently sized tubes, approximately 30 centimeters in length. They're composed entirely of smooth muscle, and they're going to move urine from the kidney down into the bladder via our peristaltic waves. If you remember from our digestive tract lecture, peristalsis is going to be the rhythmic contraction along a tube to push substance down it. So it's going to push the urine through the tube of the ureter via those peristaltic smooth muscle contractions. The urinary bladder has two parts. It has the fundus, which is the body, the upper portion, and the bladder neck, which is going to be the bottom portion. The bottom neck is where we find the triad, which are the two ureter openings and the opening of the urethra. Urine leaves the body through the urethra and exits from either the urethra opening in the female, which is its own hole, or through the urethra opening of the male. So as I stated previously, the urination process is the process of actually creating and synthesizing the urine that is done within the kidney. And these are done by the functional units of the kidney that are known as nephrons. When our nephrons are going to filter the blood plasma, they will produce the urine fluid. And that fluid is going to be known as tubular fluid. Then it is going to exit out into this area known as the renal pelvis. The renal pelvis is then going to dump down into the ureters. So the newly synthesized urine is going to move into the ureters through the renal pelvis. Again, our micturition reflex, this is going to be the process of actually eliminating that stored urine from the bladder. So urination, process of making urine within the kidney, all right, nephron synthesizing urine here. Process of eliminating urine from the body is going to be the micturition reflex. So here's the microanatomy of the bladder. All right, we are going to have this area at the top, and this is going to be the fundus. The fundus of the bladder is the top of the body. The bottom is going to be the neck. You see here in the middle, we have the trigone, which I mentioned earlier, the triad between the two urethral openings, our ureter openings, and the urethral opening. All right, so our two ureter openings here the urethral sphincter here opening down into the urethral tube. Lining the bladder itself is going to be a layer of smooth musculature. This is known as the detrusor muscle. And the detrusor muscle is going to be what contracts to help push that urine out of the bladder into the urethra. At the top of the bladder, we have the peritoneal lining. So this is going to be the bottom portion of the connective tissue lining or membrane of the peritoneum. Connecting to this substance above it, connecting to the connective tissue above it, the bladder is suspended in space by the median umbilical ligament. Down here, we can see that we have two various sphincters for the urethra. We have the internal urethral sphincter, which is going to be involuntary smooth muscle. And we have the external urethral sphincter, which is going to be voluntary. All right. Our voluntary external urethral sphincter, we can control this and allow it to open when we need to micturate. There are three layers that we see here. There's an inner epithelial lining. This is where we have transitional epithelium, one of the only places that transitional epithelium exists in the body. These cells are going to alter their shape based off of the volume inside of the bladder. These transitional epithelium are on top of the first layer known as the lamina propria. And this is going to combine to form the mucosa of the bladder, or the inner portion or inner lining of the bladder. Next, we have the submucosa, all right? That's going to be a lot of connective tissue as well as blood vessels, and also a lot of lymph flow and nerve innervation. Then we have the musculature of the detrusor muscle, and then we have an outer capsule layer known as the adventitia. So our bladder is composed of that outer adventitial layer that is going to surround the smooth muscle layer with our longitudinal visceral smooth muscle fibers of the detrusor. The detrusor muscle is going to appear somewhat striated, so it actually appears more like skeletal muscle than many other smooth muscles do. So it's going to be a little bit difficult for muscle cells to transition here. This is suggesting that the muscle cells are under their own innervative power, or at least they have the ability to innervate themselves and other types of visceral smooth muscle. Our musculature is going to surround that inner mucosal layer, and that is going to be composed of our transitional epithelium, which we mentioned earlier. This is going to allow for stretching and compression so that we can accommodate a larger fluid volume and expand the surface area inside of the bladder. All right, the process of the micturition reflex. Again, this is elimination of stored urine, actually losing urine to the outside of the body. You're going to get constant flow of urine from the kidneys down into the bladder. We have a constant drip through the ureters that is sending fluid down into the bladder. As the bladder fills, the internal portion of the bladder is going to become pressurized, and that is going to progressively stimulate afferent sensory nerves more and more for the greater fluid volume that comes in. Those signals are going to be integrated and are going to signal an efferent response if that is enough stimuli. This is called the micturition reflex. It's going to stimulate our neurological need to empty the bladder. The urethral sphincters, the internal urethral sphincter will relax. The reflex is modulated by our higher conscious nervous systems like the brainstem. Our conscious control of this can override the reflex, so we can contract the external urethral sphincter, close it off, and make sure that we do not urinate until we want to. The nerve innervation is coming from the pudendal nerve, and the pudendal nerve is going to be what allows us to contract the external urethral sphincter. All right, so our micturition reflex, we have two phases. Number one is going to be the storage phase or the filling phase. This is going to happen while we have urine accumulation in the bladder. We see the detrusor muscle in the inner lining and the transitional epithelium. As the bladder fills with urine, the transitional epithelium will flatten out and become spherical and will allow for more space to be filled. Once we get to a point where the bladder is full and those signals are being sent afferently to the brain to tell us that we need to urinate or we need to micturate, we will have an emptying phase. This is the voiding phase. This will happen when the internal urethral sphincter is relaxed and we consciously externally relax the external sphincter. The detrusor muscle will contract and push the fluid out of the bladder down through the urethra. So for our storage phase, filling of the bladder is going to be allowed by the contraction of the striated sphincter known as the somatic innervation. Okay, contraction of the smooth muscle sphincter, which is sympathetic innervation, and inhibition of the detrusor muscle, which is also sympathetic innervation. For the emptying or micturition reflex, this is going to be relaxation of the striated sphincter, right, which is somatic innervation, relaxation of the smooth muscle, and the opening of the bladder neck, which is sympathetic innervation, and detrusor muscle contraction, which is parasympathetic innervation. So during the filling phase, we are consciously aware of the fact that we are trying to contract the striated sphincter at the external urethra. This is why we are somatically innervating this. It is voluntary. The other musculature activities are involuntary. We have no control over them. And they are largely modulated by the sympathetic nervous system, except for in the case of the detrusor muscle, which is parasympathetically innervated. One of the few cases where a parasympathetic innervation is going to actually excite the process and cause something to contract. All right, so here's our conscious control mechanism. We have sympathetic nervous system afferents that are going to be sent from stretch receptors on the bladder surface. We have central nervous system efferent signals, which are going to bind to beta adrenergic receptors, and the neurotransmitter that we use here is norepinephrine. We are also going to bind to alpha adrenergic receptors on the smooth muscle of the internal sphincter, again, using norepinephrine as a neurotransmitter. The pudendal nerve is going to be going to our external urethral sphincter. And as I mentioned previously, the pudendal nerve is going to allow for somatic or voluntary control. So since it is voluntarily controlled, we have acetylcholine as the neurotransmitter here. And that is going to attach to nicotinic receptors. So our conscious control is going to go up through the brainstem. We're going to send central nervous system impulses up there. Then once we have a strong enough stimuli, we're going to send impulses back down the sympathetic chain, have sympathetic nervous system signals go to beta and alpha adrenergic receptors for the detrusor and the sphincter, right? In the case of relaxing or filling, this is going to be the reverse signal. So norepinephrine is actually going to bind to the beta adrenergic receptor, which is an inhibitory receptor, causing the detrusor muscle to relax. Also during filling, we have the alpha adrenergic receptors on the internal urethral sphincter being bound by norepinephrine. Alpha adrenergic receptors are stimulatory. So this is going to cause the internal sphincter to contract and to close. As you can expect, during the emptying phase, this is reversed. So instead of detrusor relaxation, we have detrusor contraction. Instead of sphincter contraction, we have sphincter relaxation. All right. So our sympathetic control is going to come from the T11 to L2. Again, this is from the sympathetic trunk. The parasympathetic innervation is going to come from the sacrum or the sacral spinal cord. If we remember about our sympathetic nervous system, it is a thoracolumbar, meaning that its nerves come from the thoracic and lumbar vertebrae. And the parasympathetic is craniosacral, coming from the brainstem and the sacrum. So during our filling, we have multiple signals that allow for the function to relax the detrusor muscle and to contract the internal and external urethral sphincters. We have the stretch receptors that are going to stimulate sympathetic nervous system responses. Sympathetic nervous system is going to be able to stimulate contraction of the sphincters and is going to relax the detrusor muscle, right? When we have the bladder wall that is pressed on with a pressure larger than 10 centimeters of water, the parasympathetic nervous system is going to stimulate the contraction of the detrusor muscle, all right? This is going to become the predominant signal sent by the spinal cord. The sympathetic nerve afferents are traveling via the hypogastric nerve from the T11 and L2 region. Efferent signals are going to originate and be carried by the hypogastric nerve. This is going to relax the detrusor muscle by norepinephrine binding onto the beta adrenergic receptor. The efferent signaling is going to travel to the internal urethral sphincter and bind to those alpha adrenergic receptors. These are going to produce contraction of the urethral sphincters, sealing off the urethra from the bladder. All right, during our emptying phase, now what we have is an override of sympathetic stimulation. Okay, so the sympathetic nervous system is now no longer the predominant roller here. The pressure on the inside of the bladder has gone above 10 centimeters of water. So now we have inhibitory effects on the sympathetic nervous system. Okay, so our parasympathetic nervous system is now going to send signals to the musculature of the bladder, the detrusor muscle. We will have acetylcholine, which is going to act as a stimulatory neurotransmitter, causing the detrusor muscle to contract. We have parasympathetic efferent signals going to the internal urethral sphincter, which is actually going to be nitric oxide. So nitric oxide is used here as a neurotransmitter, which is actually going to dilate and relax that sphincter. Again, we have the pudendal nerve innervation here. So we can consciously control the pudendal nerve input. And if we diminish the pudendal nerve input, this is going to diminish the stimulus to that muscle of the external urethral sphincter, causing the sphincter to relax, which will open it up and allow for a flow of urine through the urethra. So again, we're going to override the sympathetic stimulation here because we have more parasympathetic activation than we can account for with the sympathetic nervous system. So again, that increase in pressure is going to begin with the activation of the parasympathetic nervous system. Those afferent signals are going to travel from the pelvic nerve to the sacral region, all right? Spinal cord, sacral 2 and 4. The efferent parasympathetic response is going to be sent down the pelvic nerve to the bladder. This is going to contract the detrusor muscle by acetylcholine binding to the muscarinic receptors, which are also known as M3 receptors. Also, we saw our parasympathetic innervation of the internal and external urethral sphincters. The internal urethral sphincter is going to be relaxed because of nitric oxide production and binding. We also have a simultaneous parasympathetic signal that is going to travel up to the pons. Here, we usually trigger a conscious awareness of the need to urinate. So this is going to be where we have conscious awareness that comes in. That awareness is going to allow us to trigger somatic signals, which are going to go down through the pudendal nerve and go to the innervation of the external urethral sphincter. Pudendal nerve is then going to release neurotransmitter acetylcholine at this point, which is going to act on nicotinic receptors. And that is going to allow for the muscle to hold its tone until micturition is allowed. So looking at the structure of the kidney, we have a couple of functional areas. The initial portion of the kidney, this is known as the hilum. The hilum is where we have the entry and exit of all the vessels that innervate the kidney. So here we have the renal artery and vein that come in. We have the renal pelvis that goes out. We also have our nerves and lymph flow that come in here. Then we have the cortex. The cortex is the outer layer here of the kidney. This is where most of our glomeruli or glomerulus exists. Then we have the pyramids of the kidney, which are each one of these individual lobes. And each one of these individual lobes is comprising what is known as the renal medulla. At the bottom of each lobe, we have a small opening, which goes into what we call a minor calyx. And the opening at which the lobe goes into the minor calyx is going to be known as the renal papilla. Each one of those minor calyces form together and combine into a major calyx. And then all of the major calyces come together and form the renal pelvis, which then exits out into the ureter. In between our renal pyramids, we have what are known as the renal columns. And here we have what are known as the interlobar arteries and veins as well. So if we take a mid-sagittal view of the kidney, this is what that would look like. Okay. One more thing of note, we're going to characterize nephrons in two ways. So the nephron is the functional unit of the kidney. This is where we actually have filtration. There are two types. There are cortical, which are going to be mostly found in the cortex, and there are juxtamedullary, which are going to be diving deep down into the medulla of the kidney. So these are characterized by their loops of Henle, which are the renal loops of the tube. The loops in the cortical nephrons are much shallower and not nearly as dense. The loops of the juxtamedullary nephrons are going to be longer and dive down much further into the renal medulla. So the major functions of the kidney, we're going to regulate body fluid, right? This is done by osmolality, okay? We know that osmolality is going to be that concentration of electrolytes, concentration of solid particles and whatever, and how it affects the movement of fluid across a membrane, okay? We're going to affect osmolality by maintaining electrolyte balance. Also, pH for acid-base balance. It's going to act as an excretion mechanism for toxic substances, such as urea, uric acid, creatinine, hemoglobin metabolic products, and metabolized versions of hormones. Kidney is also able to produce its own hormones. We have renin, which is going to act in the renin-angiotensin-aldosterone system. This is going to monitor blood pressure and electrolyte concentrations. We have calcitriol, which is a metabolite form of vitamin D. And we have erythropoietin, or EPO, which is going to stimulate the bone marrow to produce more red blood cells. All right, so blood flow into the kidney is going to come from two vessels. We have the renal artery coming in, and we have the renal vein going out. Blood flow to the kidney is going to be very large. We're going to have about 20 to 25% of the cardiac output at rest going to the kidney, which is about 1 to 1.25 liters per minute. So we're going to start with the renal artery. As we enter in to the hilum of the kidney through the renal artery, we are now going to segment, right? And we turn into what are known as segmental arteries. The segmental arteries are going to move through the lobar portion of the medulla. And as they do, they are termed interlobar arteries. Once we get to the top of the medulla, they start to crest across the area here on the top of the pyramid. And those crests are known as the arcuate vessels, or arcuate arteries. Then they extend up into the cortical space. As they go into the cortex, they are now termed the interlobular artery. The interlobular arteries are going to split off into afferent arterioles. And the afferent arterioles are what is going to go into the glomerulus. So the afferent arterioles will enter into the glomerulus where the actual blood plasma is filtered out. Inside of the glomerulus, we have those glomerular capillaries, which are going to actively move blood plasma and exchange contents with the tubular fluid space. That arterial blood is going to exit through the efferent arteriole out of the glomerulus to the other side of the circulation. It's then going to move through the peritubular capillaries, back down through the interlobular veins, into the arcuate veins, back down through the lobes and between the lobes and the interlobar veins, and then out of the kidney back towards the inferior vena cava through the renal vein. So this is the blood flow at the level of the nephron. We have the afferent arteriole, which is going to be coming off of our interlobular artery, going directly into the glomerulus. After we go through the glomerular capillaries, that blood is going to exit through the efferent arteriole. Efferent arteriole is then going to follow the tubular flow down around the loop of Henle and back up. And this is a structure known as the vasa recta. right? This is going to be a highly concentrated blood with a lot of electrolytes and nutrients and other substances. As we move back up the vasa recta to the other side of the circulation, we are then going to enter into the interlobular vein, and the interlobular vein will take it back into the interlobar vein and back down through the renal vein. So we have our afferent arteriole, which is going to bring that arterial blood into the glomerulus. We have the efferent arteriole, which is going to take the blood away after the plasma has been filtered out. And then we are going to enter into the venous side of the circulation through the interlobular vein. All right, so that flow is going to go into the glomerulus. The glomerular capillaries are capillaries themselves, okay? So they do not turn into venules there. They exit through the efferent arteriole, not into a venule. So here's why we have afferent before efferent, okay? So afferent first into the glomerulus, efferent after once we leave the glomerulus. A portion of the peritubular capillaries are going to follow the nephron, and this is known as the vasa recta, as I saw in the image here. Again, this is an area of high solute particle concentrations that is going to loop down with the loop of Henle and back up into the interlobular vein. Right, here's the differences between our types of nephrons. Again, as I mentioned earlier, we have the cortical nephron and we have the juxtamedullary nephron. Let me zoom this in a little bit. Okay, so let's see if I can take it any further. That's close. Okay. All right, here's our cortical nephron on the left. Again, it's not very deep. The loop of Henle here is going to go down a little bit and then come right back up. So it doesn't dive very far down to the medulla. So the concentrating ability for this nephron is not that much. This is going to be 80% of the nephrons that we have. So 80% of our nephron population is going to be the cortical nephrons that we see here. The vasa recta, or the distribution of those blood capillaries that goes down, is not very far because the loop does not extend very far. So the differences are going to be how deep and dense the peritubular capillaries go and how deep the loop goes. So we can see the peritubular capillary network here is going to be pretty shallow. In the juxtamedullary nephron, however, we have a very deep diving capillary network and a very deep diving loop of Henle for the tubular fluid. And this is where we see the vasa recta come in. So, as I mentioned earlier, cortical nephrons are about 80% of our nephron population. Juxtamedullary nephrons are about 20%. So for the renal circulation, when we go from the afferent arteriole into the glomerulus, right? This is a high pressure capillary bed. It's going to filter out the fluid from the capillaries. So this is blood plasma. This is what we know as the peritubular capillary system. Okay, the peritubular capillary system is going to be after it exits via the efferent arteriole. The efferent arteriole is now a lower pressure. So the glomerulus is a higher pressure. Then once the blood exits through the efferent arteriole, the pressure is going to become much less. And this is going to cause the fluid to filter into the capillaries. This is functionally important, because in the glomerulus, we want the fluid to go out of the capillaries into the tubular space. In the peritubular capillary system, we want the fluid to move back into the capillaries from the tubular space. So having a higher pressure at the glomerulus forces the flow of fluid out of the capillaries. Having a lower pressure in the peritubular capillary system causes fluid to filtrate back into the capillaries. Pressure is going to go from about 100 millimeters of mercury in the arcuate arteries of the kidney down into about 8 millimeters of mercury in the veins, which are going to drain out the nephron. So we have a huge pressure difference there. The major resistance vessels of the nephron in the glomerulus are the efferent and afferent arterioles. The two major divisions of the peritubular capillary network are the cortical capillary system and the vasa recta, where we see the deep dive into the medullary system. Glomerulus is going to be a high pressure bed, okay? And it's going to cause the movement of fluid out of the capillaries. That filtration is going to go into the first part of the nephron, which is Bowman's capsule. Peritubular capillary system, low pressure. This is so we can pick stuff back up into the blood from the fluid of the tubule kidney, right? Again, pressures range from 100 down to 8 millimeters of mercury, depending on if we are on the arterial side or venous side. Two major divisions of the peritubular system. We have the cortical capillaries and the deeper juxtamedullary capillaries, or the vasa recta. So this is the kidney flow. Again, this is looking at a kidney that has been extracted, and we have the glomeruli filled with latex here. So each one of these individual buddings, right, is going to be where we have glomeruli existing. So we can see that there are millions of glomeruli and millions of nephrons here inside of the kidney. All right, so here's a micrograph. It's going to look pretty close into what we have here. This is a glomerulus. So these little knobs here are all glomeruli, right? Each one of these has a glomerular capillary network inside, okay? And we see that on both sides of the glomerulus, we have flow. On one side, we have the afferent arteriole. On the other side, we have the efferent, okay? So we have the afferent arteriole, for example, here's one. We have an afferent arteriole going into the glomerular space, glomerular capillaries, and we have an efferent arteriole leaving from the other side. At the top here, we have the example of a peritubular capillary network, right? And then at the bottom, we have an interlobular artery. So we can see that there is a glomerulus along the lines of the artery all the way up and down. So they're almost like stations of blood that exit here into the glomerulus as we move along. All right, so here's the nephron structures. Again, we start with the glomerulus. We start with Bowman's capsule, which is also known as the glomerular capsule. It is receiving blood flow from the afferent arteriole. As that blood is filtered out into the Bowman's capsule space, we now send down the fluid filtrate into the proximal convoluted tubule. After we go through the proximal convoluted tubule, we now go into the loop of Hindley. There's a descending portion, and then there's an ascending portion. The ascending portion is going to end in what is known as the thick ascending limb of the loop of Hindley. After we get out of the thick ascending limb, we go into another tubule known as the distal convoluted tubule. Distal convoluted tubule is going to end or cessate at a collecting duct, which is going to take the tubular fluid, which is now the urine that is produced, and go and take it back down into the calyces so it can go into the urine and out through the ureter. So we can see the top portion, the glomerulus, and most of the tubules are going to be in the cortex of the kidney. Our loop of Hindley is going to dive down into the medulla and is going to come back up into the cortex before it enters into the collecting duct. All right, so our major structures: Bowman's capsule, proximal convoluted tubule, proximal straight tubule. Those are the major structures of the cortex. Major structures of the medulla: the descending limb of the loop of Hindley, the thin ascending and thick ascending limbs of the loop of Hindley, distal convoluted tubule, collecting tubules, collecting ducts. Then we get out of the tube of the pyramids of the medulla, and we go into the calyces through the renal papilla. And those dump down into the renal pyramids, which turn into the renal pelvis and the ureters. All right, so let's label a couple of things here. We got 1 through 10, so let's go through and label these, all right? Number one is going to be the renal artery. or RA. Number 2 is going to be the renal vein, right, or RV. 3 is the ureter, or URTR here. 4 is going to be a minor calyx, right? So we're looking at 4 here, minor calyx, all right? It's going to empty down into a major calyx. 5, this is going to be an interlobar artery. 6, these are going to be interlobular arteries and interlobular veins, right? 7, interlobar veins. 8, interlobular veins. 9, we have arcuate veins. 10, we have arcuate arteries. Okay, so we're looking at the blood flow moving through here. All right, overview of kidney function. Again, it is going to be filtration of the blood. So we're filtering the plasma of the blood out. We have the afferent arteriole bringing blood in. The pressure is going to push fluid and other solutes out of the capillaries into the tubular space. We're going to filter that substance out into the tubules, which are going to either be reabsorbed, secreted, or excreted. So they're either going to be taken back up into the bloodstream, or they're going to be secreted in and out of the tubules, or they're going to be lost altogether in the urine when we micturate. So we filter substances across the glomerulus. We're going to reabsorb substances in the correct environment. So we have possible reabsorption, possible secretion. Again, this is going to be dictated by hormonal influence and as well as body fluid state. Finally, the excretion is going to come out, right? The excreted waste substance in the form of the urine. Here's our flow from the glomerulus. Again, we have the capillary lumens here. We're going to have particles leaving out from the capillaries. They're going to go through the endothelial cells there. All right. These are going to be sinusoidal capillaries, or very heavily fenestrated capillaries. They have large pores, which can allow for large particles to move out into the tubular space. We have flow come in from the afferent arteriole, leave from the efferent arteriole, and that tubular filtrate is going to be going down into the proximal convoluted tubule after it exits the Bowman's capsule. We have a couple of different cell types that exist here. At the entrance and exit of the glomerulus, we have what is known as the juxtaglomerular apparatus, or the JGA. Here we have a couple of different cell types. We have what are known as mesangial cells. This is the mesangial matrix, and we have mesangial cells there. We have what is known as the macula densa, which is going to sit here on the distal convoluted tubule. And this juxtaglomerular apparatus is going to go a long way in dictating blood pressure readings as well as electrolyte readings from the body. So we are going to comprise the glomerulus of the afferent arteriole draining into the capillaries. Then we're going to move out of the glomerulus via the efferent arteriole. Between those two arterioles are those extraglomerular mesangial cells, which I mentioned, and the distal convoluted tubule, which is going to be the macula densa. The mesangium is another tissue type there. It's thought to support the vascular tissue and release vasoactive agents when stimulated by changes in blood pressure or arterial pressure. So we have that afferent arterial flow. We have the tubular capillaries inside of the glomerulus. We have podocytes, which are covering the capillaries. They are going to act as a barrier to filtration. We have the glomerular endothelium, that is going to be fenestrated, large pores or large fenestrations, which are going to allow for substances in that plasma fluid to be lost to the tubular space. All right, so to detail this, for any substance to move from the glomerular capillary into the Bowman space, it has to cross all three of those barriers. It has to cross the endothelium through the pores and the fenestrations. It has to cross the All right, we have large fenestrations or large pores here, so everything is going to be able to pass pretty easily unless it's an extremely large blood cell. The second is going to be the basement membrane, all right, of the capillary endothelium. This is known as a visceral epithelium or the podocytes. Those podocytes are going to add an additional barrier to filtration. Lastly, we have to pass through the basement membrane. And the basement membrane is the most restrictive. It has the highest level of negative charge there. So the third barrier is going to be the podocytes or foot processes of the podocyte cells. Filtration slits are going to be interdigitating, okay? So the podocytes allow water and solutes to cross into Bowman's capsule space. All right, so those capsules here, we have our pedicles or foot processes of the podocytes, which are going to allow for filtration. If we look on the inside for the endothelial cells of the capillary, we can see our fenestrations or our pores in the lining of the capillary that fluid and substance can move through. Our filtration barrier here, we have the fenestrations, we have the pores, we have the basal lamina, or what is known as the basement membrane, and we have the filtration slits through the foot processes or the pedicles of the podocyte. So 3 layers of filtration there. All right, here's a micro view of a glomerular capillary. We have the podocyte here. We can see a primary foot process here. We have branching pedicles or foot processes here. So this is looking at an electron micrograph of the glomerulus. See the podocytes surrounding the capillaries. We have the small fern-like processes which appear there, and these are known as the pedicles. All right, again, here we have the glomerulus, we have the capillary lumen. Here, we have the endothelial cell lining. Here, we have a podocyte slit right there. We have a podocyte foot process and podocyte cell here covering this capillary. All right, so looking at the structure again, we have the proximal convoluted tubule down through the loop of Henle into the distal convoluted tubule into the collecting duct. All right, so let's identify the structures here. Number 1 is going to be the proximal convoluted tubule. Number 2 is going to be the glomerulus. Number 3 is going to be the distal convoluted tubule. Number 4 is going to be the afferent arteriole. Number 5 is going to be, actually, I think, yeah. 5 is going to be the glomerulus. 2 is actually Bowman's capsule, or the capsule space. 6 is going to be the collecting tubule. 7 is going to be the efferent arteriole. 8, we have the loop of Henle. 9, we have the descending limb. 10, we have the ascending limb. 11, we have the collecting duct. All right. So 6 was the collecting tubule. 11 is the collecting duct. All right, labeling those out. Let's see what's next. All right, so our basis of function here, again, it's to clean or clear the blood plasma of unwanted substances. So we have to get the things out, metabolites, excess quantities of electrolytes, foreign substances like drug metabolites and organics as well. All right. Finally, for the functionality of the kidney, again, we have to filter, but we can also secrete hormones. We have the ability to secrete erythropoietin, which is going to be hormonally available to produce red blood cells from the bone marrow. If we have a case of low oxygen in the blood or low binding to hemoglobin, this is hypoxia, and this will induce erythropoietin release. We also have the final activation step for activated vitamin D, which is calcitriol, right? This is going to be through a 1-alpha hydroxylase enzyme. And that enzymatic conversion is going to result in the active form, which is 1,25-dihydroxycholecalciferol. Also, we can potentially create glucose in the kidney from amino acids. So if we have a very low energy availability for the body, we have the ability for the kidneys to actually create glucose from amino acids that are being moved through the urine. So that is a gluconeogenesis process, and that is similar to the gluconeogenesis process which occurs in the liver. All right, everyone. That is the renal and urinary system. Thank you all for joining me for this lecture. Like and subscribe to the channel. Also, patreon.com/twistedscience for additional videos and content if you want to sign up there. Thank you all, and I will see you the next time for our next video.